All right. Okay, we're going to get going. Today we are moving into the world of fungi. We're going to spend this lecture and the next two lectures talking about fungi, and then move into plants and then animals. In case you forgot, there's a midterm later today. Go to the correct room assignment, please. There's a test with your name on it waiting for you, with a Scantron already in it. A through SAC in Freeborn, SAN through Z in Chem 179. And I thought, you can get out your clickers. We're going to do some practice questions related to the test, but that you can get a few bonus points here for. Please be quiet. For these ones not to be discussed among your neighbors. This is just for individual practice here. If you want to whisper, fine, I guess, or look up something on the web, I don't care. But that won't be allowed in the test. All right, so here's question number one. I'm going to give until about 45 seconds or so. All right. I'm going to shut it off. Everyone ready? All right, good. Most of you got it correct. Chlamydias are only known as parasites of other organisms. All the other answers are incorrect in some way or another. They're not autotrophic. They're not eukaryotes. This is a eukaryotic group, and there are ones that are pathogenic to humans. So in general, for the test, we're not trying to trick anyone. We do frequently put in some answers that you should be able to eliminate reasonably quickly in many of the questions. Even if you don't know the correct answer, we do sometimes put in things, like sister group to chlorophytes. If you know they're bacteria, you don't have to remember what chlorophytes, where they sit per se, to know that chlamydia are not sister group to chlorophytes. All right, here's another one. Again, about 45 seconds for this. All right. Hurry up. Let me know. Okay, good. So you may not remember that I mentioned this maybe once, that cyanobacteria are also known as blue-green algae. But all the other groups are eukaryotic groups that we talked about in a variety of ways throughout the rest of the lectures. So you should be able to recognize those, and even if you didn't know what blue-green algae were, you should know that the other ones are major groups of eukaryotes because we talked about them in a variety of ways. All right, last one. All right, so we won't do this one. Yeah, it's okay. You'll survive. Which of the following groups is not known to contain photoautotrophic taxa? You probably recognize some of them. You may not remember the photoautotrophic stromatophiles per se, but chlamydias, they're all obligate intracellular parasites that are heterotrophic. None of them are autotrophic and certainly not photoautotrophic. All right, here's the last practice one. Yeah, this should be D. But hint, it doesn't matter. All right, hurry up. All right, stopping. All right, the correct answer is B. And again, if you don't remember the exact details of all of the groups, apicomplexans are all obligate intracellular parasites. They're all single-celled organisms. And all the other groups have example of multicellular organisms. Okay, that's your little prep for the test. All right, so now we're going to switch into discussing fungi, but keep out your clicker. because in 1 second we're going to do another clicker question about fungi. So we're going to go through and talk a little bit of introduction about them, talk about the diversity of form just briefly seen in fungi, and then talk about their phylogenetic diversity. And we won't get into the symbioses. That'll be in the next class. All right, so this relates to the material we covered previously, but it is also relates to the material we're going to talk about today. All right, everyone ready? Answers all in? All right, that is the correct answer. And I will show you why in more detail. So one of the major groups of eukaryotes, of the 7 major lineages that we introduced a couple of lectures ago, is called the epistheconts. And the epistheconts includes 3 major subgroups. The animals and choanoflagellates we'll talk about later in the course when we cover animals. Choanoflagellates are very interesting. I mentioned this before. They are the sister lineage to animals. There are a lot of interesting studies that have now been done on representatives of choanoflagellates to try and understand early events in animal evolution. And the group we're going to focus on in the epistheconts is the fungi. And so an important thing to remember is that the fact that fungi are a little bit more closely related to animals than they are to many of the other eukaryotic groups is important for a variety of reasons. One is that experimental studies of organisms in this group have a little bit more relevance to animals than experimental studies of some other eukaryotic lineages. And in addition, some of the studies of fungi also have relevance to sort of understanding early evolutionary events. So if you look at this clade, like with many of the other major eukaryotic lineages, the groupings were determined largely by molecular and phylogenetic trees of sequence data. In many cases, ribosomal RNA genes, but also analysis of a diversity of other gene sequences that showed pretty clearly that choanoflagellates are the sister group to animals and fungi are related to them. Once you have that information, people have identified a synapomorphy for this episthecont group. It's not a very strong synapomorphy. As you can see, they sort of caveated here. The flagellum, if present, is single and posterior being in the back. So not all members of this group have flagella, but if it is present, it has this particular pattern. There are 2 really important synapomorphies that have been identified for this branch over here. That is the branch leading up to the common ancestor of all fungi. And we'll talk about these in more detail. They are a particular feeding lifestyle called absorptive heterotrophy and the presence of chitin in the cell walls. As I've said before, with many of the other synapomorphies we've talked about, biology is messy. There are representatives of fungi that have thrown away, chitin in the cell walls or done something weird with it. But in general, this is considered a synapomorphy. Most of the members of this group have absorptive heterotrophy, and chitin in the cell walls. So I thought since this is, test day, we would have a few movies along the way. Let me just plug in the sound here. They're about as exciting as you can have for movies about fungi. I'm just going to pause this for a second. This is an excerpt from This is an excerpt of a film made by this very well-known photographer, Louis Schwartzberg, who specializes in ultra-high resolution and super either slow motion or super sped up motion photography of nature. He's got a really interesting TED Talk. He's come out with a movie sponsored by Disney about bird and bat flight. And he also has one that's come out. This is just an excerpt of it about fungi. These are super sped up.
Mushroom mycelium represents rebirth, rejuvenation, regeneration. Fungi generate soil that gives life. The task that we face today is to understand the language of nature. My mission is to discover the language of nature of the fungal networks that communicate with the ecosystem. And I, in particular, believe nature is intelligent. The fact that we lack the language skills to communicate with nature does not impugn the concept that nature is intelligent. It speaks to our inadequacy of our skill set for communication. We have now learned that there are these languages that are occurring and communication between each organism. If we don't get our act together and come in commonality and understanding with the organisms that sustain us today, not only will we destroy those organisms, but we will destroy ourselves. We need to have a
That's not a fungus.
Consciousness.
I don't know why it's here.
If I die trying, and I'm inadequate to the task to make a course change in the evolution of life on the planet, okay, I tried. The fact is I tried. How many people are not trying? If you knew that every breath you took could save hundreds of
This one
lives
isn't
in the
either.
future, every outcome of this path of knowledge,
It's still cool.
wouldn't you run down that path of knowledge as fast as you could? I believe nature is a force.
All right. One of the reasons I like to show this is that with plants and animals, I think people have a great appreciation of both for animals, their behavior and their mobility, and for plants, certainly some of their dynamic nature, and lots of us plant our own plants and do something with them. Many people are a bit less familiar with the sort of dynamic nature of various types of fungi, and so it's sped up a little bit, but it's still pretty amazing to see the growth of these individual examples. And I just thought before we got into the biology of them, I would show this one other little video. This is another example of a fungus, sort of highlighting some of the diversity within this group of organisms. This is a predatory fungus that actually lassos, creates a little loop and waits for nematode worms to crawl into that loop and then strangles them and eats them.
Most fungi are decomposers, but the soil-dwelling fungus Arthrobotrys is better classified as a predator. The fungal hyphae form rings just large enough to fit nematodes that live in the soil. If a nematode enters a ring, the ring constricts, trapping the nematode. Hyphae then invade and digest the nematode.
I know, poor nematode. Anyway, I just thought I'd show those two things because they highlight some aspects of fungal lifestyle that people aren't quite always sort of familiar with. So now we're going to talk a little bit about general general biology of members of the fungal group. And one of these things that is really interesting in the group and is a synapomorphy for the group is this mechanism by which they acquire nutrients. And so it's this process where fungi will secrete out of their cells enzymes that can chop up proteins and carbohydrates and other compounds. And they secrete this into their environment, usually just their local environment, that degrades some material that is in their environment, say a leaf that's fallen off a tree. And then once the material is broken down, they bring the broken down products into the cell. So they don't eat by engulfment like amoeboid forms do, as we saw for many of the other eukaryotes. They don't eat like we do with some behavioral-based process. They consume largely by this absorptive heterotrophy. Many of the members of this group use this absorptive heterotrophy on organisms that are dead. So if you do this on dead organic matter, you're called a saprobe. There are others that are mutualistic symbionts, and we'll see examples of this. They are still absorptive heterotrophs, but they're getting some aid in the process of producing the broken down products by a partner, some mutualistic association. And then, as we will also see, there are many parasitic and pathogenic members of the fungal group, and there they are feeding upon a living organism as opposed to a dead organism.
In this sequence, saprobic fungi and bacteria decay bread and fruit.
So this is highly sped up, but it's just showing the way they work, and there are bacteria that do this too. Again, secreting enzymes to basically liquefy your food, and that allows you to bring in the broken down products.
These organisms, which thrive on decaying matter, fill a vital role as the major decomposers in our ecosystem.
And then they're going to show a black mold growing on bread. Looks like my fridge, so, Don't look if you're grossed out by, yucky food. It's fun to do this at home. All right. So this is a common strategy across a lot of fungi to go find some dead organic matter, or really, a fruit is sort of living and dead, right? It has the potential for its seeds to produce life. The cells are sort of still functioning, but it's not connected to the main plant anymore. But they'll also feed on any organism that dies. They rapidly get colonized by fungal spores and other microorganisms. So what we're going to do for just a couple of minutes is introduce you to two different general approaches to the, quote, form that fungi take. Basically, the choice is single-celled lifestyle or multicellular lifestyle. And so in the single-cell lifestyle, we have a group of, we have a descriptive term that is used to describe single-celled fungi they're called yeasts. You may be familiar with Saccharomyces cerevisiae. That's the yeast used in many bread baking, as part of bread baking, and also to produce beer and wine. And if you look across the fungi, you see that there are many individual representatives or subgroups within the larger groups of fungi that are single-celled, that fit this definition of free-living, single-celled fungi, that is, they are yeast. This is a homoplasious property. There are many distinct examples of the origin of the single-celled lifestyle in different branches in the fungal group. So it's not a taxonomic term. It's a description of sort of the morphology and the lifestyle. Most examples of yeast live in either incredibly moist environments or actually liquid environments. And they don't have, as you will see, these branching structures that we've talked about before as being one of the common features of a lot of fungi. But they do absorb nutrients right across their cell wall, and they are, in essence, absorptive heterotrophs. Another thing about yeasts is that many of them are really easy to work with in the laboratory, and they have become a really good model system for studying the biology of eukaryotes. So the alternative is to be multicellular, and the multicellular fungi have some similarities across all of them, as well as some differences. And I'm just going to highlight here the general structure before, when we talk about individual groups, we will see examples of how they differ from each other. This is a picture showing the inside of the xylem circulatory system of plants. That's the part of the circulatory system that's moving water around. And there's a fungus growing in the middle of the xylem circulatory system. These little branch structures are called hyphae. I'll come back to them in a second. This is pretty common. Fungi cause lots and lots of infections in plants. And if they grow a lot in here, they can clog up the xylem circulatory system and prevent circulation and either kill a certain region of the plant or kill a whole plant. If you zoom in on these structures, in pretty much any multicellular fungi, they look something like this. These are generally called hyphae. They're these branched structures that form the multicellular bodies of the multicellular fungi. They have cell walls around the outside that are made up of chitin, and they come generally in two main forms. And the forms differ in basically by whether or not, or the extent to which you have separations between the different nuclei of the cells inside these hyphae. So in one type of structure, and again, we'll see different fungus groups have different percentages of these structures, but in this type of structure, there are these things called septae. That's basically a cell wall in the middle of these hyphae. So a cell will divide as the hyphae is growing. Two nuclei will be present, and then this septae will form between the two of them. The septae don't completely close off, so you can still get nutrients, organelles, and even the whole nucleus can migrate between these different compartments, but it controls the flow a little bit compared to these other types of fungi, which you get duplication of the nucleus, but no wall forming in the middle of the structure. So these ones with the septae are called septate, and the ones with no divisions are called cenocytic. And you will see in the text and in some of the discussion of the different groups of fungi, these two terms used. And they do create some differences in the biology of these organisms because of the constraint, in essence, placed upon movement within the ones with septae as opposed to the ones without. A key feature of hyphae is that it allows for the creation of high surface area to volume ratios. Similar to many other biological features where inside the mitochondria you see all these folded over membranes inside the mitochondria. That's to create extra surface area. In the inside of eukaryotic cells in particular, there are all these different compartments like the endoplasmic reticulum and the Golgi apparatus. They're folded over membrane structures. The brain, the circulatory system in animals. There are hundreds to thousands of examples of where branching structures are used to create higher surface area to volume ratio. And that's a really important aspect of the lifestyle of these absorptive heterotrophs that increases their ability to bring in nutrients from the environment after they have either digested something or a mutualistic partner is providing it to them. This creates a problem also for many fungi in that higher surface area leads to increased loss of water in dry environments. So they are much more susceptible to desiccation than they would be if they didn't have this high surface area. And this is part of the reason why you're much more likely to find fungi in very moist, if not completely wet environments, as opposed to very dry environments. Now, there are some fungi that have figured out ways to grow in drier environments, but most of them are very prone to this desiccation. And again, why you see them. So like the black mold that grows in many buildings will show up much more frequently when the drywall gets wet than when the building is completely dry. All right, and then the multicellular fungi that you see are made up of structures that are composed of networks of these hyphae. So these come in different forms. We will see examples of this. But for example, many fungi grow in the asexual part of their life cycle as these sort of what are called mycelia, a collection of the hyphae. If they're asexual, it's also called vegetative. And this, if they grow underneath, just underneath the surface of the soil, they can serve as basically like roots, as an anchor structure for the sexually, the sexual reproduction phase, which is these fruiting bodies like a mushroom that we will see, that we saw the video of and we'll see later. This is also made up of hyphae. It's called a fruiting body, and the vegetative part here serves as an anchor. So if you look at any multicellular fungi, the different kinds of them, they're basically always composed of different arrangements of these hyphae to make up the different structures that we will see.
These time-lapse sequences show mushrooms growing.
I don't think that this has any scientific value, but it's just here, so I'll show it to you. More growing mushrooms.
Is a mycelium that lives in the soil.
Oh, it shows the spores, though, on the bottom, you'll see.
Spores are released from gills on its lower surface. The mycelium of this oyster mushroom lives within the wood of the tree it is decomposing. As it matures, the plaleus of this dung fungus curls in on itself to better expose the spore-producing surfaces.
All right, so I remembered why I wanted to show that. The mycelium of that one that's growing on the tree, so this is very common for many fungi. They will infect a tree or another plant even when it's not dead. They will be slowly killing them in many cases. And you can't even tell, except for the cases when the fruiting bodies sprout out of the tree and then grow on the outside. And that's why you'll see in all sorts of places, all of a sudden after a rain or after some other change in the environment, a lot of these mushroom-like structures coming out of the bark of a tree. That means that the inside of the tree is infected with some mycelium from that particular species of fungus. Yeah? How are fungi different from slime molds? Well, that's a good question because that's exactly what we're going to show you in the next few slides. So can you wait a second? All right, so I'm just going to do this perfect lead-in for this next section of the class here. So we've talked many times in the class about organisms that were misclassified as fungi. This was largely because of the branching structure that they took in multicellular life forms, that they had structures that looked a lot like hyphae or looked a lot like fruiting bodies that are seen in fungi. So here's an example. Do people remember what this is? These are the oomycetes, one of the lineages of stromenophiles, and it includes the causative agent of sudden oak death and potato late blight. And if you look here, or I'm not sure how well you can see it here, they have these structures that look quite a lot in a simple microscope, at least, like the structures seen in fungi. All of them are also absorptive heterotrophs. So they have a lifestyle that is a lot like fungi. That is probably why they have been selected to have these high surface area to volume ratio branching structures. But if you actually look inside of these structures, they have a very different general structure than the hyphae that we see in fungi. So they're clearly phylogenetically distinct. They don't have chitin. They have a lot of other differences compared to fungi, but on the outside, they have some similarities. So this gets to your question. Can you guess what these are? Yeah, right. So these are plasmodial slime molds, which also had undergone a lot of selection for high surface area to volume ratios. They form branched structures. These don't quite look like hyphae in many cases, especially when you look close up in them. But the Physarum species were misclassified as fungi for a long time, in part because of their life cycle and the formation of these fruiting bodies that they form. And so until people started to do more detailed molecular sequencing of these groups of organisms and then build evolutionary trees based upon core universal genes for which they had the sequences of that varied between taxa, like ribosomal RNA and others, it was hard to tell exactly what these were. But now that they have been classified as members of the amoebozoan group and into their own subgroup, looking in more detail inside their cells, looking in more detail at the actual structure of these branched forms that they take, they're very distinct from what you see in fungi, other than in the general outward appearance. They also do not, so I took out the video here, I should have probably left it. They don't feed generally by absorptive heterotrophy. Most of them feed by endocytosis. So if you observed their feeding, you would probably have rethought the classification, at least at some level. Anybody remember what these are? Right, so these are bacteria that form, some of them form these multicellular branched structures, the actinobacteria. They were also misclassified, many of them, as fungi for many years. Again, if you look in fine scale detail on the inside of these, they're really different. I mean, they have peptidoglycan in the cell wall. They have a very different type of structure. They don't have nuclei inside the cells. I mean, this is a misclassification that happened largely before people looked inside the cells of organisms in detail. Once people started to look inside the cells, it was pretty clear that these were something different going on because they didn't even have a nucleus inside their cells. Okay. So in the last few minutes, I just want to start in on fungal reproduction and some of the phylogenetic groups that we see in fungi. We're not going to get through all of them here, but we'll continue on them in the next group. So when people have built evolutionary trees of the different fungi that they've found in a variety of places, classified them into individual lineages within the fungi, and then looked at each of those groups in more detail, one of the key distinguishing features between the different groups has been the general modes of reproduction that you see in the groups. And this is going to be a common feature of what we talk about in much of the rest of the class. The distinction between many of the different groups of plants, for example, relates in a large part to differences that they have in their reproductive life cycles. And that same is true for the differences among the different the different fungal lineages. So one thing that they all basically have in common, or most of them do this, if fungi is running low on food, is starting to starve, one thing that many of them do will be to undergo sexual reproduction, or they can also generate spores by asexual reproduction, as we'll see in a little bit. And one of the reasons to do this is the spores allow for dispersal across large distances. That is not as easily possible by just growth of the mycelia. So they can keep growing, and the mycelia, there are mycelia of fungi, as we'll talk about in the next lecture, that can be tens of miles long for a single organism. But that's very slow, usually, compared to dispersing spores through the air. And so many fungi have specialized mechanisms to project spores up into the air to allow them to get better dispersed by wind. This is a puffball. I'll show you a little video of them in a second. This is another type of fungi that has basically a water cannon-like dispersal process, and it disperses its spores in order to get them away from its local environment and hopefully get picked up by, actually in this case, cattle. And amazingly, there are other organisms like worms that have figured out that they can hitch rides on these water cannons of these fungi by crawling up onto them right before the spores go off, and that's what this video shows.
But as cows don't eat their own muck, how do the spores of these fungi get into the cow?
You'll see the spore growing in a here, this one. So it's going to fill up with water, and then it projects these little spore packets.
The spores at its tip are sat on top of a miniature water cannon that fires the spores beyond the ring of grass that has grown up around the cow pat and that the cow won't graze. The spores that bombard the pasture
And they're really sticky,
will only be dislodged when the cow
so they
eats and
stick to wherever they land.
grass.
And these are the worms that then crawl up and hitch a ride on these fungal sort of water cannons.
In their life cycle. What better than to hitch a ride on a jet-propelled fungal artillery shell?
And then here's a little video of these puffballs, which usually are triggered by things like rain dropping on them.
Produce spores within the fruiting body. As with most fungi, the spores are dispersed by wind. When
So,
a drop of rain hits
this
the
is in
puffball,
slow motion.
spores are forced out through a small opening, giving them a boost for better dispersal.
So dispersal of spores is a really important part of the life cycle of various fungi. And when people have compared the different groups of fungi, they've also noticed some key similarities or differences both in the asexual phase of the life cycle and the sexual phase of the life cycle. So I'm just going to go through, we're going to go through these again when we talk about the individual groups. You should remember, so one of the key things about the asexual phase of the life cycle here is to remember that mitosis is the process by which eukaryotic cells make copies of themselves, right? So a eukaryotic cell makes a copy of itself by mitosis. It turns out that for fungi, for many groups of fungi, they form structures, sort of bodies in both the haploid phase and the diploid phase of the life cycle. That's not like animals. So our haploid phase does not form a whole body. It just is there for sexual reproduction. But in fungi, as we'll see, they can have sort of multicellular structures in the haploid and diploid phase. And in each of those phases, the organisms grow by mitosis. And then they can produce gametes from the diploid phase by meiosis. So you'll see in the life cycle diagrams that will show some parts of the life cycle, there's reproduction just by mitosis, and then in some phases there's reproduction by meiosis. If you look at the sexual reproduction in eukaryotes, in fungi, that we'll see, and we'll give examples of this, in some of the groups, some of the major lineages of fungi, there's no known sexual reproduction. That is, all reproduction is by asexual means. It's still considered possible that it might exist for some of these lineages, but it has never been detected or observed. Fungi also don't have males and females in the same general way that we talk about male and female for animals, and as you will see, also for plants. There is no obvious morphological distinction between the different partners that engage in the sexual reproduction in fungi, but there is a molecular distinction. So what you have in fungi is what are called mating types. So you have a population within one species. There's a region of the genome, sometimes just one gene, that is polymorphic, that has variants in the population. And to prevent inbreeding, what happens is that you can only reproduce with a member of a different mating type. That is, that has some differences at this one region of the genome. And in essence, it's a way of preventing inbreeding. And in some species, there are 2 mating types. In others, there are dozens. And you always have to find some organism in a different mating type in order to undergo sexual reproduction. And we'll see when we talk about the life cycles how this works in different lineages of fungi. So I'm not sure how familiar everybody is with what's called alternation of generations, but I thought I'd just go through it quickly here. So many eukaryotes, plants, for example, as we will see when we talk about plants, switch between a haploid phase of the life cycle and a diploid phase of the life cycle. They spend roughly equal, amounts of time or equal amounts of effort in the haploid phase and the diploid phase. That's certain plants, not all of them. If they spend roughly equal amounts of time in these different phases of the life cycle, it's called alteration of generations. And so the process, is the process that should be familiar. The terms here were terms that will come up when we talk about plants. But a diploid cell will undergo meiosis to produce haploid cells. Those haploid cells in alteration of generation actually grow to form a multicellular organism. In these plants, it's called a gametophyte. Don't worry about that. We'll come back to it. But they actually reproduce by mitosis to form a multicellular organism. And then eventually, they produce gametes, also by mitosis. And those gametes are haploid. That is, they have 1 copy of each chromosome. They will fuse by fertilization to produce the diploid offspring. That will then grow as a multicellular organism, and the cycle goes through again and again and again. So that's called alteration of generations when you have multicellular organisms in both the haploid and the diploid phase. In animals, the generations are very biased, such that the haploid phase does not have a multicellular organism. The multicellular organism is only seen in the diploid phase, and that's called a diplontic life cycle, that bias towards the diploid phase. And then you see some organisms where the opposite of this is seen, where there's a haploid phase with a multicellular organism, and it's the diploid phase that is basically trivialized, such that there's no real multicellular organism in the diploid phase. This is called a haplontic life cycle, with the haploid phase being dominant. And we see some of the fungal lineages have this particular life cycle. So all sexually reproducing eukaryotes go through this alteration between haploid and diploid phases. The extent of time and whether or not they have multicellular individuals in both of these phases determines whether or not we're going to call them alteration of generations, where both the diploid and haploid have multicellular individuals, diplontic, where just the diploid has the multicellular, and haplontic, where just the haploid has the multicellular. And so if we look at fungal life cycles, again, with that as the background, one of the main comparisons that distinguish the different groups of fungi are in exactly how they divide up the haploid versus diploid parts of their life cycle, and then the processes by which they merge different lineages to produce the diploid, and the processes by which they generate haploid cells. This is just a generalized life cycle. You don't have to worry about the details here. So I'm just going to do 1 or 2 minutes on the major groups of fungi, and then we will talk about them later. There are 6 major lineages that have been identified of fungi, at least as outlined in the book. Again, like all the eukaryotic groups, like the bacteria and archaea, there are other lineages here that are not being highlighted, that are mostly poorly understood, but there are other groups. Microsporidia, chytrids, zygomycota, glomeromycota, ascomycota, and basidiomycota. Unfortunately or fortunately, it looks like the group that is called chytrids may be polyphyletic. That is, there may be multiple branches separating out from the base of this tree that all lead to what are called chytrids. We're going to treat them for most purposes as, potentially a monophyletic group, but you should sort of remember that it's unclear if they are for certain. The same is true of the zygomycota. There appear to be potentially multiple lineages that lead to what are all called zygomycota, but we're going to treat them in essence as a monophyletic group. You should learn these groups, should learn what's in this tree, and then there's a table that highlights the key features of these groups. You should learn what's in that table, table 30.1 in the book. And good luck on the test.